Hear me? All right, cool. So thank you everyone for coming out today. I really appreciate it. Thank you, Scott, for that introduction. I did not regret too much sharing the screen. Everyone here has been in some way a huge help to this project. So I just want to open up with a huge thank you to everyone that's on this call. And there's a handful of people that couldn't make it, and thank you to them as well. Before we get started, just a quick reminder to please keep yourselves muted. Have as much fun as you want in the chat, go off in there, and then I will answer questions towards the end. So without further ado, let's get going. This is the title of my talk, my dissertation, A Hopeful Monster in Aquilegia, or perhaps more specifically, uncovering the genetic basis and selective advantage of a naturally occurring floral homeotic mutant of Aquilegia caerulea. And yes, before anyone asks, this is going to be a Zach Cabin story that's got a lot of background information and excruciating amounts of detail, so buckle up. We're going to start where every evolutionary biology talk should start, with Darwin. So Darwin's idea of evolution by natural selection postulates that slow, gradual phenotypic change over geologic time creates the patterns of diversity that we see today. Now this, with the rediscovery of Mendel's work, and an increased understanding of genetics and applied mathematics and statistics, brings us to the modern synthesis in the 1930s. And the synthesis states that if the mechanism for phenotypic change is slow and gradual, then so too is the mechanism for genetic change. Small effect micro mutations slowly fine-tuning an organism to their environment. The argument being most populations and most organisms are either at or near their fitness optima. So a large effect mutation is much more likely to overshoot that target. Now on the complete other side of the spectrum, we have what's called saltational evolution. And saltational evolution states that the diversity we see today is mainly driven by, or at least partially driven by, evolutionary leaps, saltations, large effect macro mutations that can create a new lineage almost instantly. Now, we can't mention saltational evolution without mentioning this guy, Richard Goldschmidt. He was an evolutionary biologist during the time of the modern synthesis, and he had some problems with this entirely gradual approach. The way he saw it was micromutations are really important for variation within species and local adaptation, leading to varieties, or a sport, as Darwin would call it. But true species, he argued, distinct units and higher level taxonomic rankings are separated by what he called bridgeless gaps. And he couldn't quite come to terms with how successive micromutations could ever span these gaps. So to explain away these inconsistencies, he invoked this idea of what he called a hopeful monster. It's a little campy, but it's fun. And the idea is that a single effect macromutation, a single genetic change of large effect, could drastically alter early development and create a radically different body plan in the adult organism. And this radically different body plan could lead to a new branch on the evolutionary tree, but it would have to occur in the proper ecological setting. Goldschmidt himself admitted a lot of these mutations would either be lethal upon occurrence or would occur at the wrong place at the wrong time. But at the right place, at the right time, with the right change, you could have a hopeful monster as opposed to a hopeless monster. Now, as developmental genetics and DNA sequencing have changed the way we look at evolutionary biology, we can ask ourselves, what do the data say now around 80 years later? And it's clear that this all falls on a bit of a spectrum, with a huge majority of evolution occurring through this gradual Darwinian mechanism. But there's definitely a place for these large effect macromutations. There are a lot of adaptively important traits that have been shown to be under the control of large effect loci. But almost all of these are then supplemented by many small effect modifier loci. So this super extreme example of saltational evolution, this idea of a hopeful monster, hasn't really been shown. Now in Goldschmidt's times, he recommended looking at homeotic mutants, or organisms where one organ is replaced with another, as evidence for these types of monsters. These are most commonly seen in the lab, like this example of Drosophila. This is a wild type head of a fruit fly. And this is a mutant where a developmental gene called proboscipedia has been knocked down, and you get a replacement of mouth parts with legs. I think this is what we would call a hopeless monster, but nonetheless, as Goldschmidt would argue, this proves that genetically, these types of changes are very possible. Small changes to a developmental patterning gene could have drastic effects in the adult. Now, unfortunately, the natural evidence is a little bit more lacking. The best evidence we have is the lack of transitional forms in the fossil record. So large jumps that can't really be easily explained by successive micromutations, segmentation in arthropods, where anciently diverged lineages are separated mainly by the number of segments on their body. And then, of course, what Darwin referred to as the abominable mystery, the apparently sudden appearance and explosion of flowering plants in the fossil record. Now, two interesting things about arthropods and flowers. In both of the evolutions of these organisms, homeotic patterning genes have been implicated as being important. And these are two of the most successful, if not the two most successful groups of organisms on the planet. So it's worth noting that they do invoke some idea of perhaps a saltational evolution or this hopeful monster change. But these macroevolutionary comparisons, although informative, are exactly that, comparative. They are really hard to imply causality. It's hard to say whether the origin of flowers led to the split between angiosperms and gymnosperms, or some other trait led to the split, and then later on flowers arose and allowed for radiation. We don't know the selective pressure that caused the split, but we do know that it is a stable transition. On the other hand, we could look at a microevolutionary example. How well do homeotic mutants fare in the field? Well, there's also not that many examples of that. This paper came out a few years ago, and they say that many spontaneous floral mutants are due to homeotic changes. An interesting question is whether such mutants could survive in the wild and whether they have the potential to ultimately give rise to novel species. Every time we see a homeotic mutant that does well in the field, these are always occurring in selfing or clonal lineages. So their effect on reproductive isolation and the ability to lead to a new lineage is really unclear. As such, this question relates to the debate about gradual versus saltational modes of evolution. Few data are available on the fate of such floral mutants. If you've seen any talk and you understand how these talks are structured, until now. So what I'm gonna do today is sort of put together this hopeful monster puzzle, if you will, where I'm going to show that a single macromutation, loss of function alleles at a homeotic patterning gene, lead to a radical phenotypic change, a replacement of petals with a set of sepals. I'm going to show that this is occurring in the proper ecological setting, and floral herbivory, not pollinators, surprisingly, is driving the shift in floral morphology. And I'm going to lastly show that this phenotype is involved with reproductive isolation and could lead to a new lineage. And finally, I'm going to stand on the shoulders of some pretty good macroevolutionary work and show that this is a very stable macroevolutionary transition that could lead to a new lineage. But before we do that, I have to introduce the study system, Aquilegia. So why do we study this group, columbines? For starters, they're awesome flowers and we get to flex on people in our talks with photos like these. But in actuality, it's because they are a great model system for studying evolution, and it's thanks almost entirely to this structure. On each of these species, I've pointed out the petal and I've highlighted it in this one species on the left. And that is a petal, but it's a highly modified petal with this tubular outgrowth we call a nectar spur. Now, these nectar spurs are really important to the rapid radiation of this group, the rapid speciation, because the length, shape, and orientation of these spurs has a really tight correlation with specific pollinators. And when a pollinator forages for nectar, it deposits pollen on the chest in a very specific spot. And in a population, if there's mismatches between these spur lengths, pollen doesn't get deposited in the right way, and you end up with reproductive isolation and speciation. Furthermore, these are really important for the reproductive fitness of these organisms by promoting outcrossing or mating between non-relatives. In almost every species of columbine that has been looked at, there's really strong inbreeding depression, which is exactly what it sounds like. The more inbred you are, the less fit you are for the environment. So from first order principles, we can say that a loss of spur is the most extreme form of a mismatch, and this should be really bad, perhaps a hopeless monster. But at the right place at the right time, maybe it has some hope. So I study Aquilegia cerulea. This is the Colorado columbine. It's a state flower of Colorado, and it is primarily pollinated by hawk moths, although bumblebees are a major visitor as well. And in our study population, it's at around 75% frequency. Now let's orient ourselves to the flowers. The first whorl of flower are these flat purple structures. excuse me, and they are the sepals. And there are 5 of those, and they alternate with these modified petals. This white part is called the blade, and that comes down and forms the spur with the nectar here at the bottom. Now, about 120 years ago, this mutant was first described, Aquilegia cerulea var. Dahliae. It's a variety technically. And it's a homeotic mutant in which the petals have been replaced with a second set of sepals. So you can see that the outermost whorl, lost my pointer. The outermost whorl are these same flat purple structures, but the inner whorl is another set of those structures. So a huge change. And in that same population, it's at around 25%. It makes up the rest of the group. This high frequency indicates that it's at least selectively neutral and may even have some fitness benefit. And when we look at a dissection of the 2 flowers, we can see that apart from these second whorl organs, there really isn't a difference in the phenotype across these plants. And that agrees with the original description. This is a drawing from the original description. And in it, Alice Eastwood writes, "This is in all respects similar to the wild type, except that the spurs of the petals are entirely wanting. Sepals and petals are flat and blue." Also in that description, she mentions it's found in 3 places. By a Miss Anna Daly in Evergreen. Daly is where we get the Dahliae name. By an Osterhout in Estes Park, and then herself in Platte Canyon. Well, this blue star is Reynolds Park. This is our study population. You can see it's just outside of Denver. And these are those 3 localities that she described. So it's not surprising that we're finding it at high frequency nearby the original descriptions. But that's actually not the most interesting part about this paragraph. She says right here that Miss Daly noted that it was in her opinion that the plants had become more numerous since she first noticed them. So even from the first description, there's been some evidence of a selective advantage, a fitness advantage for this group, for this morph. So we ask ourselves, how is this mutant surviving, #thriving, in a natural population? Well, our first question requires us to just observe what's going on, put on our ecology hats. And that's what we did for 3 consecutive field seasons, 2014, 15, and 16. 2015 with the help of Konstantin Gazelholm, and 16 with Tim No. And the first thing we would do every single year is we would create a spatial map of the population using this laser-based triangulation setup we came up with. That was all the lasers you saw in Scott's videos. So we would make this spatial map, see where every plant was relative to each other. And then we would tag every single flowering plant. We would mark its morphology. This is a scan from our field notebooks. We would mark the morphology, numbers of flowers, any types of herbivory on the plant, anything of note we would write down. And then we would come back in about a month or so during the fruiting season and record whether or not any particular flower set a fruit, created seeds into the next generation. This was our measure for reproductive fitness. Did they create a fruit? For the flowers that did create a fruit, we would take a couple seeds for genetic analysis, and we would also take leaf tissue off of every single plant for genetic analysis as well. I can't talk about fieldwork without sharing some fun photos of us doing pollinator observations, all of the fun hikes and really cool field sites we got to go to, all the places we got to eat lunch, the places we got lost, and all the people's whose houses we stayed at. Luckily, it was 3 seamless field seasons. Nothing bad ever happened. No one ever lost any field notebooks. No one ever crashed any cars or almost got in a little bit of running with bears. It was great. Nothing happened. It was all thanks to this car. We've been faithfully dubbed the Mobsda because we mobbed around Colorado in it, put around 20,000 miles on this car over 2 summers. Really just a beast, best off-roading car on the market. And yes, if you're asking, those side doors are sliding doors like these. But at the end of every season, we would have one of these spatial maps where we could see every single flower relative to everything else. The blue dots are Dahliae plants and the red dots are wild type. And that color scheme will continue throughout the entire presentation. In case I can't convince you with this map, here's a photo. These plants are growing right next to each other. This appears to be a completely naturally occurring common garden, which is ideal for us because we can now study the differential fitness effects of these morphologies under theoretically the exact same selective pressures and environmental variables. So after 3 years of measuring fruit set, did we notice any difference in reproductive fitness? And the answer is a resounding yes. And I'll walk you through this graph. Each set of columns is a different year, 2014, 15, and 16. And these numbers on the bottom are the total number of flowers for that year. The red bars are wild type, blue bars Dahlia. And the y-axis is the proportion of flowers that in this case successfully set a fruit. And the asterisks indicate significance. The more asterisks, the more statistically significant the comparison was. Across all 3 years, we see a huge, consistent fitness advantage for the Dahlia flowers. They are much more likely to successfully set a fruit than wild type. And this is really surprising given the assumed disadvantage we have given the morphology change. So we have to ask ourselves, what is driving this pattern? And the first question we want to look at is spatial differences. We think this is a common garden, it looks like one, but maybe there's some fine scale geographic variation, differences in soil chemistry, sunlight availability. Essentially, we have to make sure, are these morphs actually evenly distributed throughout the population? Or is there some non-random clumping that could be driving the difference in fruit production? So what we would do is we would take the population, these maps we made, and we'd break it up into 16 evenly sized quadrats. And we would take a random flower, essentially ask, what is the probability this falls in any one of these 16 quadrats? In other words, are the distributions of these morphs different based upon a randomization test? And the end result is a graph like this. And without getting into the details, each light colored gray line is a summary function for an individual quadrat, and the colored lines are the mean summary functions. And since the slope on those two is the same, we can say, no, there's no difference in spatial distribution within these populations for the two morphs. Now, another question we could ask is there a resource allocation issue? Maybe different flowers, numbers of flowers per plant, different fruit sizes, seed numbers. Is it possible that making a spur is that energetically costly that the Dahlia plants, by not making one, can make larger flowers or more flowers? So the first question we asked is, are they making more flowers? So across all 3 years, we looked at the total number of flowers per plant. You see here on the x-axis, and again, the proportion of plants on the y. And you can see just by looking at the lines, there's no difference, but we have our statistics from a Mann-Whitney ranked sum test. There's no difference in the flowers per plant for either morph across all 3 years. So that's not driving the difference in fruit set. Now, what about fruit size? Right now, we're just using a binary variable, yes or no, did they set a fruit? But what if Dahlia plants are able to make much larger fruits? That would extend their fitness advantage. On the other hand, what if wild type fruits are a lot larger? Maybe they're setting less, but they're bigger. This was actually Constantine's idea in 2015, and then we implemented it with Tim in 2016. So what we would do for every fruit is we would measure the height and width in 2 directions and model the volume off of an ellipsoid. And first, we collected unopened fruits from around the population, not within that specific map that I showed you. We would count the total number of seeds in there and then see if there's a correlation between seed number and fruit size. And there's a really tight correlation. As fruits get larger, they have a lot more seeds. So we can use fruit size as a proxy for seed number and actual total reproductive output and ask, are there a difference between the two morphs? The answer is no. What we're looking at here is the distribution of fruit sizes between the morphs. And as you can see, the distributions are almost exactly the same. There is no difference in fruit sizes and seed number by that matter between these morphs. So it doesn't appear to be a resource allocation issue. Now, some of you may notice that there is quite a lot of year-to-year variation in overall number of fruits and proportion of fruits that set flowers that set fruit. And yes, totally fair, there is. But I would like to bring your attention to the fact that the magnitude of this difference is relatively consistent across all 3 years. Dahlia is consistently better at setting fruits than wild type plants. And it seems as though this is likely driven by climate, where specifically temperature and rainfall. 2016, our worst year technically for fruit set, was by far the hottest and driest year that we were in the field. In 2015, the best year technically was the most temperate in terms of temperature, as well as the wettest. But again, the important thing here is whatever is driving this, it is affecting the morphs equally, not differentially. So that's not driving the difference in fruit set. So then the last thing we land on is pollination, the culprit we would assume it would be. And I just want to show this video to show hawk moth pollination, to show how deep into the flower these pollinators get when they're foraging for nectar, and to explain how close in contact with the reproductive organs that they are. You can see it's really getting into like the neck and body of the hawk moth. So you can see already there's a huge amount of mechanical isolation just created by this lack of nectar spur. On the other hand, there are also bumblebees, and bumblebees are probably moving a lot more randomly because they're not foraging for nectar, they're foraging for pollen. But we have to first ask ourselves, are both pollinators even visiting both morphs in the population, or a Dahlia or wild type losing out on one pollinator? And specifically, are they transitioning between? So we set up GoPro cameras in 2014 on multiple patches of flowers, and we were able to capture some evidence of hawk moth and bumblebee visitation. Oops. So this is for hawk moth visitation. There we go. And you can see that a hawk moth comes into the bottom corner right here, then actually tries to feed on a Dahlia plant, which is surprising because its tongue has nowhere to go, just kind of pokes it, and then moves to a wild type. Now, if you look at bumblebees, we can see that it visits a wild type, it then visits a Dahlia flower, and again, a wild type. So this is just a subset of the data. This is all of the data sort of summarized. The purple arrows show hawk moth visitations, the gold arrows show bumblebee. It's not a lot of data, obviously, but it's enough to say that both pollinators are visiting both morphs, and they appear to be transitioning between the morphs as well. So our next question is, can these flowers set fruit without pollinators? And the answer is yes, but with an asterisk. A lot of work has been done in the pollination ecology of this species by Russell Miller and Joanne Brunet. And they've shown that these are self-compatible, but when you exclude pollinators, if you cage a flower, the self flowers have significantly lower seed production, so smaller fruits. And they've also shown that if flowers are open pollinated, essentially not manipulated at all, then the first flower, the flower that develops in the first flower position, the fruit that develops from that tends to be larger and the most likely to set fruit. So we have to ask ourselves, with this information, can we control for flower number and any disturbances like herbivory, essentially give a plant every opportunity to successfully set a fruit? Is there a difference between the morphs? So our null hypothesis is that if Dahlia plants aren't receiving adequate pollen, we would expect them to be making smaller fruits and these fruits to be less likely to set fruit. Excuse me, less likely to set fruit. The way that we did this was we compared only flowers that made the same number of flowers per plant. So one flowered wild types to one flowered Dahlias, 2 to 2, 3 to 3. And then we also made sure that none of the flowers on the plant had any forms of herbivory or any types of disturbances. The idea being the only remaining variable that could affect fruit set would be pollinator visitation. And we asked two questions. Are first flower fruits different sizes for undisturbed plants of either morph? And then are either morph more likely to set fruit in the absence of a disturbance? So for that first question, are the first flower fruits different sizes? This is the results. And this is the same type of graph I showed you a moment ago with fruit sizes, but we have it divvied up by the first flower fruit from one flowered plants, the fruit that developed from the first flower position in 2 flowered plants, and then again in 3 flowered plants. And again, looking at this, you can tell that between morphs, there's not a huge amount of variation, but between years, or excuse me, between plant size, essentially plant number, flower number, there is. And if we put this through an ANOVA, you can see that morphology has no effect on the fruit size of these fruits, but FPP, or flowers per plant, does, where 2 and 3 flower plants make much larger fruits. This is likely indicative of some type of resource availability limiting one flower plants. But again, the take home here is there's no difference between the morphs. No. Now our second question is either morph more likely to set fruit in the absence of a disturbance? Well, we did this across all 3 years. We compared flowers per plant, FPP, for 1 flowered plants. We would then take all of the Dahlia flowers that set fruit or didn't, and wild type that set fruit or didn't. And then we would put them through a Fisher's exact test and get a p-value. But we did this for 1 flower, 2 flower, and 3 flower plants for 2014, 15, and 16. So 9 total comparisons. So these p-values are adjusted for the multiple comparisons. As you can see, only 1 comparison, 3 flowered plants in 2014, barely crosses our threshold of significance at 0.05, and 3 flowered plants are very rare. So honestly, no. In the absence of disturbance, it doesn't seem to be a difference of any of these morphs to set fruit. So what this means is that pollinators are not affecting whether or not these plants are setting fruit, nor the size of the fruits. Now, they could be affecting seed quality. We know that as hawk moth abundance increases, outcrossing rate increases as well. And we don't see that relationship with bumblebees. So if Dahliae are losing hawk moth pollination, could an increased amount of bumblebee plus a decreased amount of hawk moth visitation lead to higher inbreeding in the Dahliae plants? Keep in mind, these plants suffer from inbreeding depression. So this might not be great for Dahliaes. So what we did is we assessed inbreeding using SNP genotyping probes at 4 neutral positions throughout the genome. These were all 4-fold degenerate, meaning they were about as neutral as we could assume a position to be. And we would genotype every single population, every single flowering plant in the population in 2016 at these 4 positions, and then split the population into 2 groups, Dahliae plants and wild type. And we would measure the F statistic, the inbreeding coefficient for those 2 groups. We would be able to get our total number of heterozygotes from the genotyping and then use the allele frequencies plus an expectation of random mating to get our expected. And notice that if observed and expected are equal, that ratio is 1. So your inbreeding would be 0. Again, the higher inbreeding, the more inbred you are. Pretty straightforward. And as you can see across all 4 loci, we have Dahliae on the left, wild type on the right. There is significantly higher inbreeding in the Dahliae plants. So not only are pollinators not driving this difference in fruit set, but they actually might be leading to a fitness cost for the Dahliae plants, which makes this fitness advantage that we're measuring here in fruit set all the more impressive. But unfortunately, it's kind of leaving us out of possibilities of what might be driving it. The only thing left is a non-pollinator agent of selection. We had a really good idea our first year of what this was, but in our second year, we really figured it out. Can you guys hear that? No, I don't know. This is a fun time where we spooked a deer on accident, but this was a really good little bit of data that we actually gathered. It's a short video, but it's really important because we had thought that deer were eating a lot of the flowers, but we could never seen it. We never been able to prove it. And capturing this video was really good evidence. Also, that little patch of flowers that this deer is eating is about 50-50 Dahliae wild type, and it only ate the wild type flowers, and that's important. Now, there are 3 different herbivores, or excuse me, across all 3 years, this herbivory pattern really seems to be driving the difference. So this is the exact same setup as the fruit set graph I showed you a moment ago, but instead of the y-axis being the proportion of flowers that set fruit, it's the proportion of flowers that suffered herbivory, and the scale is a little bit different. But again, a consistent difference favoring the Dahliae plants. Across all 3 years, they're significantly more likely to set a fruit and significantly less likely to get eaten. Now, there are 3 herbivores. The first is aphids, small little sap-sucking insects that tap into the phloem of the inflorescence and suck out nutrients. Caterpillars, identified by Ashley Quezada as Platypolia anceps. And these bore through the sterile tissue, the petals and sepals, and eat the reproductive organs. Not great for fruit set. And then, as you saw, deer, which completely removed the flower from the plant. Aphids likely affect fruit set because when they get really encrusted, they sort of cause the flower to wilt and really suck up some nutrients and might affect visitation. Caterpillars eat the carpels, and the anthers, which are not good for your reproductive fitness. But both of these herbivory types can still result in a successful fruit set. Deer herbivory is 100% penetrant. Every single flower that suffered deer herbivory did not set a fruit. It's perfectly correlated. So we know this is driving our difference in fruit set, some combination of these 3 herbivores. But the question is there a preference? So we were able to do a logistic regression where we did morphology as the outcome variable and the presence or absence of different herbivores as the independent variables. And these are the odds ratios. An odds ratio of 1 indicates there's no preference. And the way it's been coded, an odds ratio greater than 1 is a preference for wild type, less than 1, a preference for Dahliae. And you can see in 2015 and 16, aphids prefer wild type, and 2016, the deer prefer the wild type. But the important thing here is nothing is ever preferring the Dahliae plants. So there seems to be consistent selection via floral herbivory on wild type plants that is favoring the Dahliae morphology. So if we go back to our puzzle, we can say that we have a radical phenotypic shift, this homeotic mutant where petals are replaced with sepals. And we can say that it appears to be occurring in the proper ecological setting. It has higher fitness. Now, I can't fully remove this puzzle piece because we've only shown selection over a couple of years at one stage of the life cycle. We need a little bit more information to be more confident. Specifically, we need genetic information. So what we need to do next is understand the genetic basis of this mutant. We know it's a homeotic shift, so we can look at homeotic patterning genes and floral development to try and get a good idea. And luckily, a lot of work has been done on the genetics of floral development. And it can basically be summarized by what we call the ABC model. This was developed in Arabidopsis, but generally holds water pretty well amongst flowering plants. And the way it works, there are these three types of genes, A, B, and C class genes are what we call organ identity genes. So where they're expressed and the combination they're expressed can lead to the development of very specific organs. And they're also all transcription factors. They are factors associated with transcription. They bind to DNA. Now, there are four whorls of a flower. The outermost whorl, whorl 1, are your sepals, and those are controlled by just A class genes. Whorl 2 are your petals, those are A and B class genes. Whorl 3, are stamen, those are your B and C class genes. That's your male reproductive organs. And then whorl 4 are your carpels, just C class genes, those are your female reproductive organs. Now we can be a little more specific, give them gene names. Notice there are 2 B class genes, AP3 and pistillata. We are concerned with AP3 today. And we can get really specific and look at the ABC model and Aqualegia specifically. And we can see that in this group and in the family, actually, there have been multiple duplications to the AP3 lineage, such that there is an AP31, AP32, and AP33. And luckily, we know going in that AP33 is highly specialized, highly subfunctionalized to the petals. You cannot make a complete petal without function of AP33. And we know this from work in Elena Kramer's lab. What they were able to do over in Harvard was knock down expression of this gene, AP33, in columbine flowers, and they get the same petal to sepal transition that we see in these Staliae mutants. So on the left here, when it says whorl 1, this is a sepal from one of the flowers where they knocked down AP33. And this is a sepal from one of our floral dissections. You can see they're pretty much identical. And this is the second whorl organ from when they knock down the expression of AP33. So a petal should be developing here, but you get this organ, and this is almost identical to the second whorl organs on Dahlia plants, down to even the attachment point at the bottom, which is slightly different from the attachment point on a true sepal. So expression, knockdown, and loss of function, phenocopies are mutants. So we have a really good candidate gene. So what we did is we sequenced 101 plants at AP33 in the surrounding regions, around 3,500 base pairs. These were a combination of field collected plants and then some seeds that we collected in the field and grew up in the greenhouse. So 81 of these plants we actually had morphology data on. And our question was, are there any alleles that are in the sequence region at AP33 that have a clear loss of function effect on the locus? Well, we identified 55 variable positions, and 4 of them appeared to have loss of function effects. This is a gene diagram showing the wild type with the black area being the black line being the sequenced region. These gray boxes are exons. The red dot is a stop codon, and then this red diamond is a known regulatory element. The most common mutation we found was this 1 base pair mutation noted by this blue triangle, and that causes a premature stop codon in the first exon. Then we found a 9 base pair deletion. This one's pretty interesting. It's entirely in frame. So the entire protein gets produced, but the 3 amino acids that this removes are highly conserved amongst flowering plants in the AP3 lineage. Now remember, these are transcription factors. They bind with DNA. And this part of the gene is the DNA binding motif. So it probably has some importance and function. We found another 1 base pair deletion that also causes a premature stop codon. And lastly, a 682 base pair deletion immediately upstream that removes known regulatory elements. So we ask ourselves, are these loss of function alleles, which we're going to call Dahlia alleles or D alleles, associated with phenotype? Well, these are diploid organisms, so they can have either 0, 1, or 2 copies of any allele. And we can see that every single Dahlia plant has 2 copies of these non-functional or assumingly non-functional alleles, and none of the wild type do. So this looks like a totally recessive phenotype, where you need both copies of the null allele in order to get the phenotype. But it's a small sample size, only around 100 plants. So we expanded to the entire population, 848 samples, every flowering plant from the field season in 2016. And we designed a few more of those SNP genotyping probes I mentioned earlier for the 2 1 base pair deletions and the 9 base pair deletion. We couldn't really design one for that 600 base pair deletion. Now, our assumption is that if the genotypes at AP33 are causing the phenotype, then and it's recessive, then every Dahlia plant will have some combination of 2 Dahlia alleles, and every wild type plant will have either 1 or no Dahlia alleles. That's our assumption. And that's almost exactly the data that we see. A huge majority of Dahlia plants carry 2 alleles, and a huge majority of wild type plants carry 1 or none. Now, there are these mismatches, like this 15 Dahlia plants that only carry 1 or none of the alleles we tested for, but we know we're missing some variation. We didn't assay at all for that 600 base pair deletion. And also in a sample size of 100 plants, we captured 4 alleles. There's likely variation at this population that we haven't quite captured yet. And these 13 wild type plants that appear to have 2 Dahlia alleles could be the result of compensatory mutations, restoring function to the gene, recombination between alleles such that one haplotype is carrying 2 alleles, but it's paired with a wild type. So it technically only has 1 Dahlia haplotype, linkage to unknown variation, or simply error during PCR or sample labeling and collection and laboratory processing. Regardless, with these mismatches, we have a really strong correlation between genotype and phenotype. So we can say with a lot of confidence that genotype at AP33 is causing the phenotype of the Dahlia mutant. But what we really want to say is that the mutations that we found were causal. And to do that, we need to look at a haplotype analysis. Now, haplotypes are essentially a chunk of DNA that's inherited directly from your parents. And for any region of your genome, you can look at all the variable positions, and you can, across the population, you can phase haplotypes and essentially see how each variable position is linked to other positions in that region. And then you can group each haplotype based off of some characteristic allele, say the Dahlia mutations. And then you can put them into a network like this, and show how all of these haplotypes are related to each other. Now, there's a lot on this screen, so I'll walk you through it. Each of these circles represents a different haplotype. The size of the circle represents how common it is in our data set in the population. And the color corresponds with the way we've grouped it, the diagnostic allele. So all of the blue haplotypes, 2, 3, 21, and 12 over here, carry the 1 base pair mutation. Now, the difference between, say, the haplotype number 3 and haplotype 21, you see there's this 1 hash mark right here. There's one other variable position, but of all the 3,500 base pairs we sequenced, there's only 1 variable position between those 2 haplotypes. And that's what each hash mark represents, a variable site. Now, if the Dahlia alleles we've identified are causal, they should be the only thing separating a functional wild type allele and a non-functional Dahlia haplotype, which is exactly what we see, for example, with the 9 base pair deletion. Haplotype 13 is a functional wild type. There's 1 change. We know that this change is the 9 base pair deletion. And then you have the 9 base pair haplotypes. So we can say that the 9 base pair is causal of the phenotype. We can say the same thing for the low frequency 1 base pair deletion right here and the 682 base pair deletion here. And I don't really have the time to get into it, so I just need you to trust me, but we can say the same thing for the high frequency 1 base pair mutation as well. So overall, we can say that multiple independent recessive loss of function alleles at AP33 are causing the Dahlia morphology. So now we can go back to our puzzle and we can say we can remove this idea of a single mutational change. We can show that it's just the loss of function allele that is causing this huge shift. Now, with this information, we can go back to our question about fitness. How long and how strong has selection been acting, is selection and how long has it been acting on this morphology? We measured really strong selection in the field from a selection coefficient from around 0.17 to 0.3, which, trust me, those are really strong for naturally occurring populations. But we only caught it over 3 years. We could have been lucky. And we only showed it at 1 stage of the life cycle at female fitness nonetheless. There is plenty of other places throughout the life cycle in which wild type could recuperate some of that fitness loss, or DALY-A could sort of extend its lead. Now, luckily, both of those patterns are going to leave distinct traces in the genome. So now that we have the genetic basis, we can look for molecular signatures of selection. The metric we're going to use is called extended haplotype homozygosity. And in a population, you can imagine that a new neutral mutation arises. When it pops up, it is linked to the entire chromosome. So the haplotype is as long as it can possibly be, but it's as rare as it can possibly be. It's in one heterozygote of the population. So really long, but really rare. Now, over time, recombination is going to shuffle it with other haplotypes in the population, making the length of the known linked region smaller. And it's going to drift around in frequency. It might get really common, it might stay rare. But the end result is this distribution in a population of low frequency long haplotypes, low frequency short haplotypes, and low and high frequency short haplotypes for neutrally evolving alleles. Now imagine a haplotype that arises that is under strong selection. Well, the vector decreasing its length via recombination is going to be the same. That's agnostic to the selective function of the allele. But the vector pulling it up in frequency is going to be much stronger. Selection's actually taking a little hand and pulling that up to high frequency. So you end up with a really high frequency haplotype that's really, really long, that we know what it's linked to really far away from the causal locus. Now, because the 1 and the 9 base pair alleles are at high frequency, we can actually look at this metric. So I'm going to show you two different homozygosity graphs on the scale of 0 to 1. This is the same data that I'm showing first. This is haplotype decay for wild type haplotypes. The only difference is where the decay is centered, on either the 1 base pair mutation, this is a gene diagram right here, or the 9 base pair mutation. Now, this pattern, what we're seeing here, is kind of exactly what we would expect for an old, neutrally evolving allele. Really rapid decay. Recombination has broken this up, so entire haplotypes are actually very short. The takeaway is the longer you can stay, your haplotype homozygosity can stay at or near 1, the stronger and more consistent and longer acting selection has been. And that's exactly what we see for the haplotype homozygosity for the 1 base pair haplotypes and the 9 base pair haplotypes. The main thing here is this pattern is exactly what we would see if these two alleles are under really strong, consistent positive selection. This means that the patterns we measured in the field were not only accurate, we didn't just get lucky, but we're likely incorporating a lot, if not all, of the fitness advantage that is gained to wild type plants by lack of herbivory. This also really follows the patterns of what we would call a multiple origin soft sweep. Essentially, this happens when the alleles that are under selection tend to be recessive or a large functional target like a loss of function mutation. So multiple functionally similar alleles arise and are all pulled up to high frequency. So the molecular signatures are sort of dampened across each locus, as opposed to a single allele that is pulled up and sort of holds on to all of the signatures. Again, exactly the patterns we would expect for a soft sweep. So we can come back to our puzzle and we can really say that we know that this is occurring in the right ecological situation. We've been able to show strong fitness at the ecological level and at the molecular level, meaning it's been happening for some time. Now, lastly, or not lastly, almost lastly, we want to see whether or not this can potentiate a new lineage. Can the DALY-A mutant lead to a new species? Well, one of the ways in which species arise is through a combination of divergent selection and a reduction in gene flow, assortative mating. Now, this is by all means not the only way that speciation can occur, but in sort of this lens of a hopeful monster and selective advantage, this is how we might expect it to work. Now, in Reynolds Park, we can't really show divergent selection. What we're showing is more of a directional selection, pushing towards the DALY-A optimum. There doesn't seem to be a huge benefit for wild type plants in Reynolds Park. But in other populations, if there's differential herbivory, there may be a huge advantage for wild type if there, let's say, was no herbivory in the population. And that can create much more of sort of a splitting up, sort of a divergence between the two groups. If we take that and add assortative mating, a reduction in gene flow, we might get a speciation event, or at least the early stages of. And we know there's going to be a huge amount of mechanical isolation and likely reproductive isolation just because of the way hawk moths forage for nectar. They may visit dahlia plants, but whether or not they are effective pollinators remains to be seen, but is unlikely. Bumblebees, on the other hand, are moving somewhat randomly. Now, that being said, bumblebees have been shown on Cerulean specifically that they learn to prefer flowers with a higher load of available pollen, because again, these guys are foraging for pollen. So if dahlias are not getting visited by hawk moths, they're going to have an excess amount of pollen, and that may result in preferential visitation from bumblebees. So you still may get some asymmetric pollen movement there. So how can we look for patterns of assortative mating with the data that we have? Well, we have a slight issue. Assortative mating is driven by pollinators that are moving pollen between specific morphologies, dahlia to dahlia, wild type to wild type. And the result here is going to be an overall lack of heterozygosity, a lack of heterozygotes. But if we're looking at a population with such a fine scale lens, so close at such a specific area, we might also run into this issue of population structure. Now, structure is driven by, pollen just moving to the closest plant, regardless of morphology. The pollinators are just going, whatever is the next plant in their line of sight is where they visit. But you tend to be related to the things in your neighborhood, especially for plants with poor dispersal mechanisms like columbines. You tend to share alleles with people you're related with, people being plants in this situation. So again, the result is a lack of heterozygotes. So we sort of have this situation where population structure and heterozygosity and assortative mating lead to the same patterns. But because we have the locus, we know AP33 drives the phenotypic shift. And we know that the phenotype is what's going to be driving the pollinator discrepancies. We actually expect a really specific type of lacking of heterozygotes, a very specific lacking of heterozygotes from assortative mating, specifically at AP33. But if it was structure, it's just going to be all heterozygotes across the board are lacking. And we can use that to kind of determine what's going on. Using our genotyping probes from before, we were able to get allele frequencies for each locus. And then we were able to look at 4 different genotypic classes: dahlia homozygotes, dahlia hets, so they carry 2 different dahlia alleles, dahlia wild type hets, so any of the 3 dahlia alleles and then a wild type allele, and then homozygous for wild type. And using our allele frequencies and our expectations of completely random mating, these are the counts of plants in the population. This is in 2016, we would expect to see. Now, I really want to draw your attention to these 2 middle rows, what we're going to call the intramorph hets. Those are dahlia heterozygotes, and the intermorph hets. Now, regardless if these patterns are driven by assortative mating or population structure, we're going to expect an excess of homozygosity, regardless. And similarly, at these intermorph hets, we're going to expect a lack of heterozygosity. Now, the really specific pattern comes to what we're going to call these intramorph hets. If the patterns we're going to observe are driven by population structure, pollinators are just visiting the nearest plant. And again, you're likely to share alleles with those around you. There's going to be some local clumping of particular dahlia alleles. So there's going to be a lack of dahlia hets. But if it's assortative mating, then the pollinators are choosing based off of morphology. They're not caring about the genotype, they're caring off of morphology. And they don't really care if it's a het or a 1 base pair homozygous or a 9 base pair homozygous. They're going to be moving somewhat randomly between those plants. So we'd expect much closer to our expectation of random mating if it's assortative mating. And that's actually exactly what we see. These are our observed values. And as you can see, we have an excess of homozygosity we would expect and a lack where we would expect. But right here, at these dahlia hets, we have about equal. These patterns much more closely match the patterns expected of assortative mating than just population structure. And this is really significant according to a chi-square test. This is very different from expectations. Furthermore, we can look at these terms called the standardized residual. And this essentially asks which of these categories contributed most and significantly to the chi-square statistic, with an absolute value greater than 2 being considered significant. Well, we have this huge standardized residual for dahlia homozygotes, and that's actually not that surprising. If you think back to earlier in the talk, we showed that dahlia plants suffer from a little bit higher inbreeding depression. So or excuse me, a higher inbreeding coefficient. So this isn't necessarily super surprising. But notice this huge lack of the intermorph hets. The main way these plants are going to be formed, not the only way, but the main way, is pollen moving between morphs. And there's a huge lack of those, meaning that pollinators are not really moving pollen efficiently between the morphologies. But again, within a morphology, regardless of genotype, we got almost exactly what we would expect. So it does look like there are patterns of assortative mating in this population, and that this phenotype is driving those patterns. So we can come back to our puzzle and remove the last sort of tenet of Goldschmidt's problem of a hopeful monster, that it could potentiate a new lineage. And lastly, what we want to look at is this a stable transition? Is the loss of a nectar reward, the loss of a petal, a pollinator reward, a viable evolutionary path? Well, we know at least one time in Aquilegia it happened. This is Aquilegia calcarata. This is the lone spurless species of columbine, and you can see it's smack dab in the middle of the phylogeny, so it's a secondary loss. Now, this is not a petal to sepal transition. This is a loss of nectar spur. It's under the control of a different transcription factor. But like the macroevolutionary comparisons I mentioned earlier, we don't really know what drove this split necessarily. We know that it's stable. We know that it's a totally safe, fine lineage, but we don't know what caused it from a selective point of view. Furthermore, within the Ranunculaceae, the family that columbines are in, there have been 9 independently occurring losses of petals. So this is a very common evolutionary occurrence within this group. 7 of those 9 occurrences implicate loss of function alleles at AP33 and being correlated with the loss of petals. I've pointed out all of the 9 here, and the red arrows are ones in which AP33 was implicated. The repetitive occurrence of the same phenotype loss of petals indicates that it was most likely in some way being selected for these lineages. So yes, we can say this is a very stable macroevolutionary transition, and we get some bonus material that it seems as though the genetic transition, the genetic mechanism, loss of function at AP33 is the preferred way to do it as well. So we can wipe out that part of our puzzle all the same. So what I've shown you here is that a single macromutation, a single genetic change, loss of function alleles at AP33, causes radical phenotypic shift, a homeotic mutant in which petals have been replaced with a set of sepals. And I've shown that this is occurring in the right ecological setting, with heightened herbivory favoring fruit set for dahlia plants. And I've corroborated this with evidence of natural selection at the molecular level around the AP33 locus. I went on to show that it is implicated in reproductive isolation and could potentiate a new species in that manner, and that this transition, the loss of petals, is a macroevolutionarily stable phenotypic and genetic shift. So all in all, this is by far the best example we have today of that extreme example of saltational evolution, Goldschmidt's hopeful monster. Thank you. That's everything. I do want to say thank you to a handful of people, all of my funding sources, obviously, for allowing me to do this work, the department for backing me up across the board whenever I needed help. There were definitely times where it felt like you guys were more worried about me meeting deadlines than I was, and that was always very appreciated. And I do want to shout out the intro bio team. They were sort of like a second little lab family that I had, and a lot of my professional development is attributed to them. I also want to give a special shout out to Tom and Todd, the members of my committee, for whenever I had a question, they were there to answer it. Whenever I gave a talk at a seminar, you guys made me think about things in a different way that definitely made the work better for what it is. So thank you. Science does take a village, though, specifically a village of undergraduates. I don't have any pictures, unfortunately, of Ashley, but I have pictures of Constantine and Tim and Alex Garton, 3 people that I get to call good friends now. But I can't stress enough to everyone watching, this work wouldn't be possible without these 3 people. It was an honor to be able to put you guys on the paper because you guys were really integral to this work. And to all of my friends sort of outside of my lab families, if you will, all my friends outside of the lab family, whether it was, friends from Santa Barbara, friends from home, you guys were a huge buoying force, making me get that one last drink at Llama Dog, or going to the beach, or going on a hike, or going golfing, whatever it was. You guys are the best. Thank you. A special shout out to a few people, though. Jackie, my girlfriend, for supporting me through all this. I met Jackie right when I started studying for quals, then through a pandemic, and then now studying for my exit seminar. So all of the mental breakdown aspects of the PhD and none of the fun parts, all the field work and visitation, but she stuck with it and supported me along the way. To my brother, Jonathan, my best friend, and my new sister, Nicole, you guys from day one have been super supportive. You guys, I said this before, you have no idea how much I look up to you both. So thank you. And of course, to my parents who across this entire journey have supported me along the way. I definitely think there were points at the beginning where they, like me, were a little confused with what I was doing, but they stuck with it. And Dad, I am done. You no longer have to have that obligation of paying for gas. I filled up the last tank of gas in the car, so you're good on that. I also want to give a final thank you to my lab family. This is the only 2 photos I could find of Jason. So he's a sneaky guy, but he has been a really fun addition to the lab. Also not pictured here is Nathan Derrig, who actually originally got me involved in the lab at the very beginning. He was my TA in Scott's genetics class, and he introduced me to this system, and I really would not be here without Nathan. So thank you to Nathan as well. And here we have our collaborators in the Kramer lab, who I unfortunately didn't get to work as close as I would have loved to, but you guys are super fun time, a lot of great people, and you guys do really cool science. And pictured here in the corner, we have the Finkelsteins, my favorite plant development people that I've ever met. They're just, you guys are the best. But I can't talk about my lab family and not mention Angie. She is sort of my non-academic advising mentor. What gave you the idea to take this 22-year-old under your wing and turn him into the scientist he is today doesn't really make sense to me, but you did it and you were there, and we went to a lot of Muni nights, and it was fun convincing the bartenders why nectar spurs were an important evolutionary development, and matching at the airport. Those were all good experiences that I'll treasure. And of course, lastly, to Scott, the head honcho, what possessed you to allow, as you said, an undergrad in your genetics class who barely got a C, let's not sleep on that, to work in your lab, and then offered him this awesome position in graduate school. I don't know, but it was a super fun time. I think I was a bit of a weird grad student at times, but I hope it was as fun for you as it was for me. This has been a life-changing experience and wouldn't change it for anything. Everyone else, just seriously, thank you so much. This has been an awesome experience. And with that, I will open it up to questions. Thank you.